Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee. Welcome to the Stack series.
It is wonderful to be back with you all.
Today, we're going to start with a bit of a paradox, really, and it's one that sits right at the very center of your existence. I want you to think about the one thing you absolutely, positively cannot live without. If I take it away for, say, 3 minutes, you're in very serious trouble. It fuels every move you make, every single thought you have.
You have to be talking about oxygen.
Exactly. Oxygen. It's the very fuel of life. Without it, our mitochondria, the little power plants in our cells, they just shut down. Energy production grinds to a halt, and, well, we fade to black.
But that's where the paradox comes in, the cruel irony.
That's it. That same element, the one that is keeping us alive second by second, is also slowly killing us from the inside out. It's like a slow burn.
It is the oxygen paradox. It's the fundamental price we pay for being aerobic organisms. You know, creatures that use oxygen to make energy really, really efficiently. The downside is that the process is, well, it's inherently messy. It isn't perfect. It creates these toxic byproducts. We are, in a very literal biochemical sense, rusting.
Rusting. That is the image, the one word I want everyone to just hold in their heads for this entire conversation. Because today, we are exploring oxidative stress, aging, and the material degradation of the world around us. And, we look at the stack of research for this one. I mean, it ranged from dense biochemistry papers to material science journals. I was just surprised by how much the whole conversation has shifted.
Oh, it's shifted dramatically. I mean, if we were having this conversation in the 1950s, or heck, even the 1990s, we'd be talking almost exclusively about wear and tear.
Right. The idea that the body is just like an old car that runs down from too much mileage.
Exactly. But the papers we have in front of us today, the ones looking at the landscape of 2025, 2026, they tell a much, much more complex story. We're moving from the simple idea of random damage to the really cutting-edge science of zombie cells.
Cellular senescence.
Cellular senescence, right. And the drugs, the actual drugs that might be able to reverse that process.
And what I absolutely love about this stack is that it's not just about human biology. We've got papers here on material science, on how plastics are degrading in the ocean, on the structural failure of polymers. And weirdly, the underlying chemistry is almost identical.
That's the beauty of it. And I suppose the terror of chemistry. Oxidation is oxidation. It doesn't matter if it's happening in your mitochondrial membranes or on the bumper of an old car. The fundamental mechanism of electron theft is surprisingly similar.
So we're going to look at both.
We have to. We're going to explore how we rust and how the world that we've built is, well, rusting right along with us.
So let's start at the very beginning, the foundations, what we think we know. We hear this term oxidative stress all the time. It's on skin cream bottles. It's in supplement ads. It's a huge buzzword. But let's strip away all the marketing. What is it, really?
At its absolute core, oxidative stress is an accounting problem.
It's an accounting problem.
It's an imbalance. It's a failure of, let's call it, chemical budgeting. On one side of your body's ledger, you have the production of what we call reactive oxygen species, or ROS, and their cousins, reactive nitrogen species, or RNS.
So the bad stuff.
The potentially bad stuff, yes. And on the other side of the ledger, you have your body's defense system, the antioxidants. Now, when the production of the ROS and RNS outpaces the ability of the defense system to neutralize them, that's when you're in a state of oxidative stress.
Okay, let's zoom in on the bad stuff then. The free radicals.
Mm-hmm.
I always picture them as these chaotic little rebels just smashing things up inside the cell. But physically, chemically, what makes them so aggressive?
That's actually not a bad mental image. But let's look at the atomic structure. A free radical is simply an atom or a molecule that has an unpaired electron in its outer shell.
An unpaired electron.
Yes. And in the quantum world, electrons are, well, they're very social. They want to be in pairs. They are stable that way. You have one spinning up, one spinning down. When an electron is all by itself, it makes the entire molecule incredibly unstable and highly, highly reactive. It becomes desperate.
Desperate for a partner.
Precisely. It will do anything to find another electron to complete its pair and get back to a stable state. And it does not care where it gets it from. It will rip an electron off of its nearest neighbor. And that neighbor could be a strand of your DNA, it could be a critical protein or a fat molecule in your cell membrane.
And this is where the chaos starts. Because when it steals that electron, the victim becomes a radical itself, right?
Yes. And that is the absolute key. It creates a domino effect. A chain reaction. I like the analogy of a frantic dancer on a crowded floor who is looking for a partner. They grab someone, leaving that person's previous stance partner all alone. Now that person is frantic and grabs someone else, and in the process, they're knocking over tables or breaking glasses. They're disrupting the entire party.
And the tables and glasses, that's our cellular machinery.
Exactly, our cellular structures. And this entire chaotic cascade happens in nanoseconds. It's unbelievably fast.
So who are the bouncers? Who stops the fight? The antioxidants.
They're the peacekeepers. Or maybe a suicide squad is an even better metaphor. See, antioxidants are molecules that are chemically unique because they are generous. They can donate an electron to that frantic free radical to satisfy it. But here's the trick. They can do so without becoming unstable themselves.
So they break the chain.
Yes, they stop the chain reaction cold. Your body makes its own incredibly powerful ones. Enzymes like superoxide dismutase, usually just called SOD and catalase. And of course, you get the ones from your diet, the exogenous ones like vitamin C and vitamin E.
So you have this constant war going on. Radicals trying to break things, antioxidants trying to fix them.
Yeah.
But this idea isn't new. This goes back, 70 years? To a guy named Denham Harman.
Denham Harman is the father of this entire field. Absolutely.
Yeah.
In 1956, he proposed the free radical theory of aging, the FRTA. And you have to understand, it was a truly revolutionary idea at the time. Before Harman, the prevailing thought was that aging was just some kind of genetic program, that we were simply designed to fade away after we reproduce.
Like planned obsolescence.
Exactly. Harman came along and posited that aging isn't a programmed destiny at all, but rather the slow accumulation of random damage over time. He argued that the very act of breathing, of metabolizing food, creates these free radicals. And over decades, that damage just builds up and builds up until the system starts to fail.
The rusting of biology.
Yeah.
But let's get really specific. When we say damage, I want to know what is actually breaking. Because the sources we looked at highlight three main targets: DNA, fats, and proteins. Let's start with the DNA because that seems the most high stakes.
It is. DNA damage is arguably the most dangerous because it affects the very blueprint of the cell. If the blueprint is wrong, everything the cell builds from that point on is going to be wrong. And the sources mention a very, very specific marker for this kind of damage called 8-oxy-G.
8-oxy-G. It sounds like a droid from Star Wars, but I'm guessing it's not that friendly.
Not friendly at all. It's short for 8-oxo-7-filiar-7-tallers dehydrotumlinonos deoxyguanosine.
Okay, let's stick with 8-oxy-G.
Yes, let's. It's actually a mutated guanine base. You know your DNA has the four letters, A, C, T, and G. Well, guanine, the G, is particularly electron-rich, which makes it a really attractive target, a magnet for oxidation. When a free radical, especially a nasty one like a hydroxyl radical, hits it, it turns into 8-oxy-G.
So the letter G itself gets rusted. What happens when the cell tries to read that rusted letter?
It gets confused. When the cell needs to replicate its DNA to make a new cell, the replication machinery doesn't recognize the G anymore. It looks different. So instead of correctly pairing it with cytosine, the C, it often mistakenly pairs it with adenine, the A. This causes a specific type of mutation we call a G to T transversion.
So it's a typo, a permanent typo baked into your genetic code, all because of a little bit of rust.
That is a perfect way to describe it. And if that typo happens in, say, a gene that suppresses tumors, you can get cancer. If it happens in a gene that controls metabolism, you get dysfunction. And the sources also point out that free radicals can do more than just change the letters. They can physically break the DNA backbone, causing single-strand or even double-strand breaks, which are just catastrophic for the cell to repair.
Wow, okay, that brings us to the second target, lipids, fats. Now, usually when we talk about burning fat, it's a good thing. But here, we're talking about the fats that actually make up the cell itself, the membrane.
Right. Every single cell in your body is wrapped in this delicate double layer of lipids. It's the skin of the cell. When free radicals attack these specific lipids, the process is called lipid peroxidation.
And this is that domino effect you mentioned earlier.
Yes, lipids are extremely prone to that chain reaction. One oxidized lipid molecule will attack its next-door neighbor, and that one attacks the next, and so on. It turns the membrane from this flexible, fluid skin into something that's stiff and leaky. It fundamentally compromises the integrity of the cell. If the membrane is the castle wall, lipid peroxidation is like the mortar crumbling to dust.
And the sources note that this kind of damage is particularly bad in the brain.
It is.
Why there specifically? Is it just because the brain is mostly fat?
Correct. The brain is incredibly rich in a specific type of fat called polyunsaturated fatty acids, or PUFAs. And those PUFAs are highly, highly susceptible to oxidation because of their chemical structure. This is a huge reason why oxidative stress is considered such a major driver in neurodegenerative diseases like Alzheimer's and Parkinson's.
And finally, proteins, the third target.
Protein oxidation often leads to a specific kind of damage called carbonylation. Essentially, the protein structure gets modified, it gets oxidized, and it starts to unfold or clump together with other damaged proteins.
Is that like an egg white cooking? It starts clear and liquid and then gets white and solid?
That's a fantastic analogy. The concept is very similar. The protein loses its three-dimensional shape. And in biology, structure is function, always. If the protein loses its shape, it can't do its job. Even worse, these damaged proteins can form what we call aggregates, these insoluble clumps of garbage that the cell has a very hard time clearing out.
They're gumming up the works.
Literally gumming up the works. This accumulation of protein garbage is heavily linked to sarcopenia, the loss of muscle mass and function in aging, and of course to those infamous plaques we see in the brains of dementia patients.
So to sum up the biological side, DNA gets typos, membranes get leaky, and proteins get gummed up. That's the picture of rusting inside us. It's a pretty grim picture. But as we promised, we're also looking at the world around us. Because this stack included some fascinating chemistry papers on polymers, plastics. And it turns out my old plastic lawn chair is suffering from the exact same problem as my cells.
It's uncanny how similar the mechanisms are. I mean, in material science, we call it polymer degradation. But the enemies are exactly the same, oxygen and energy.
And in the case of my lawn chair, that energy is coming from the sun, right? UV light, photooxidation.
Correct. Think about a piece of plastic, any plastic, left out in the sun day after day. The UV photons, these little packets of energy, are bombarding the polymer chains. That energy excites the electrons in the plastic molecules, kicking them into a higher, unstable energy state. And this creates, you guessed it, free radicals right there inside the plastic matrix.
And just like in the body, those newly formed radicals want to react with oxygen in the air.
Immediately. They react with atmospheric oxygen to create what are called peroxy radicals. These peroxy radicals are aggressive and they attack the polymer backbone, that long, tangled chain of molecules that gives plastic its strength and flexibility. This leads to a process called chain scission.
Cutting the chain.
Literally snipping the chain. The long, spaghetti-like strands that make plastic durable and flexible get chopped up into shorter, weaker segments. This is precisely why old plastic gets brittle. It's why it yellows and cracks. Its structural integrity is being snipped away at the molecular level.
The sources mentioned a term I'd seen before but never understood. Chalking. You see an old vinyl siding or, patio furniture. You touch it, and this white powder comes off on your hand.
That powder is the degraded, oxidized polymer on the very surface of the material. It has literally rusted away and detached from the main body of the plastic. And this connects directly to a massive environmental issue that our sources brought up, microplastics.
Right. We tend to think of microplastics as just small pieces of plastic that got ground down somehow. But it sounds like they're actually the direct result of this oxidative failure.
That's exactly right. The photooxidation makes the bulk plastic brittle. Then mechanical force waves in the ocean, wind, friction, can easily shatter that brittle material into trillions of microscopic fragments. So, in a very real sense, microplastics are the shed skin, the debris of material oxidative stress.
And there's also heat, right? It's not just UV, thermo-oxidative degradation.
Yes. Heat does essentially the same thing as UV light. It provides the activation energy needed to break chemical bonds and create radicals. Heat plus oxygen equals chain scission. It's a universal problem for all organic materials, whether that material is a strand of your DNA or a polyethylene grocery bag.
So we've established the mechanism. It seems very clear. Oxygen, energized by metabolism or sunlight, creates radicals. Radicals break chains, they mutate codes, things fall apart. It seems pretty straightforward.
It does seem straightforward. And for a very long time, that logical simplicity led us to a very specific conclusion. A conclusion that seemed inescapable. If radicals are the bad guys, we just need to flood our system with the good guys, the antioxidants, and we'll stop aging, maybe even live forever.
Which brings us perfectly to part 2, the active debate. Because, as you said, it turns out it is not that simple at all. I vividly remember The antioxidant craze of the 90s and early 2000s, vitamin E, vitamin C, beta-carotene, people were popping these huge megadoses thinking they were buying an elixir of youth. It was a billion-dollar industry.
And it was the logical assumption based on Harman's theory. This is what we now call the antioxidant paradox. The hypothesis was simple. If free radicals cause aging, then removing them should stop aging. It's just simple math. Right. But then scientists actually ran the massive, expensive, long-term clinical trials, giving thousands of people high doses of vitamin E or vitamin C for years, and the results were, frankly, shocking.
They didn't work.
It was worse than that. Not only did they often show no benefit whatsoever, but in some very well-known cases, they actually increased mortality.
Increased it.
Yes.
Huh?
There were trials and some large meta-analyses referenced in our stack where people taking high-dose vitamin E had a statistically significant higher risk of certain cancers or even heart failure compared to the group taking a placebo.
That is just, it's huge.
Yeah.
And completely counterintuitive. How can something that neutralizes the bad guys end up being bad for you?
Our sources point to a few key explanations. The first one is a problem of logistics, bioavailability and location. Taking a pill is a very, very blunt instrument. When you swallow a vitamin C tablet, it has to survive your stomach acid, get absorbed into your blood, and then somehow get inside the right part of the right cell.
Specifically inside the mitochondria where most of the radicals are being made.
Exactly, and the damage is happening incredibly fast. A free radical, like the hydroxyl radical, is one of the most reactive chemical species known. It reacts in nanoseconds. It hits the very first thing it sees. A vitamin molecule just floating around in the cytoplasm of the cell is simply not fast enough or in the right place to stop a radical that is generated right next to a strand of DNA inside the nucleus.
It's an X problem.
It's absolutely a kinetics problem. The defense is too slow and too far away to stop the attack.
So the pill can't get to the fire in time. But the sources mention another, more personal reason, right? Something about our individual genetics.
Yes, this is crucial. Patient heterogeneity. Our sources discuss that everyone's internal or endogenous antioxidant machinery is different. It's based on your genetics, and we're now learning, even on your specific gut microbiome. For some people, their built-in system, the enzymes their body makes, like glutathione peroxidase, is already highly optimized. For them, adding a flood of external supplements might just overload the system or interfere with other delicate biochemical processes. So one person's potential cure is another person's useless, or in some cases, harmful supplement.
But here is the part of the research that really just blew my mind, the plot twist. It turns out we might have been completely wrong about ROS being purely bad in the first place.
This is the concept of hormesis, and it's a critical, fundamental shift in our entire understanding of this field. For decades, we viewed reactive oxygen species as nothing more than toxic waste, just exhaust fumes from the cellular engine. But biology is rarely that wasteful. It's incredibly efficient. And it turns out the body uses these radicals. They are signals.
The sources use the example of exercise, which is just a perfect illustration of this.
It really is the best one. When you exercise, you are burning fuel at a very high rate. Your mitochondria are working overtime. You are creating a massive amount of oxidative stress in your muscles. Now, according to the old theory, this should be unequivocally bad. It's a huge burst of damage. But we all know exercise is one of the best things you can do for your health. Why?
Because that burst of stress is a signal.
Exactly. The sudden spike in ROS acts as a messenger. It's a signal that travels to the cell nucleus and essentially says, hey, we are under a heavy load out here. The engine is struggling. We need to build more and stronger mitochondria. We need to make the muscles stronger. We need to ramp up our own internal antioxidant enzymes to prepare for the next time.
So the damage itself is what triggers the repair and, more importantly, the upgrade.
Correct. That is the definition of hormesis, a beneficial adaptation to a mild intermittent stressor. But here is the kicker, and this has been shown in multiple studies now. If you take high doses of antioxidants like vitamin C and E during or immediately after your workout, you block that signal. You mop up all the ROS before they can deliver their vital message to the nucleus.
You mute the signal, so you do the whole workout, you sweat, you put in the effort, but you don't get the full physiological benefit.
You blunt, or in some cases completely block, the training adaptation. You lose the hormetic response. This single realization has shifted the entire field. We're moving away from the free radical theory of aging and towards what some of the newer papers are calling the signaling disruption theory.
I really like that distinction. So aging isn't just about accumulating rust. It's about a communication breakdown.
Think of it like trying to listen to music on an old radio. The ROS, in the right amounts, are the music, the signal telling the cell what to do. But as we age, two things happen. First, the background static gets louder and louder. That's the accumulation of random unrepaired damage. And second, the volume knob gets broken. The cell becomes deaf to the signal. It stops hearing the music. It stops knowing when to repair, when to divide, or even when to die. The noise just becomes too loud for the cell to function properly.
That is a profound shift in thinking. It means our goal shouldn't be to just kill all the radicals. We need to somehow restore the proper signaling balance, bring the static down, and fix the volume knob. And that leads us perfectly into part 3, the frontiers. Because if the cell stops listening, sometimes it enters a state that is, well, it's pretty terrifying.
We are now talking about cellular senescence.
Zombie cells.
Ah.
This is my favorite part of the stack because it genuinely sounds like science fiction, but it's happening in, my knee joint right now as we speak.
It's a catchy name, but it's scientifically accurate in a very real way. A senescent cell is a cell that has permanently stopped dividing. It has reached the end of its line. This usually happens because its telomeres, the little protective caps on the ends of its DNA, have gotten too short after too many divisions. Or because it has suffered so much DNA damage from that rust we talked about earlier that it's at risk of becoming cancerous.
And normally, a cell in that state should just die, right?
Yeah.
A process called apoptosis should fall on its sword for the good of the organism.
Exactly. Programmed cell death. It's a critical safety mechanism. But these cells don't die. For complex reasons, they resist apoptosis. They enter the state of suspended animation. They just linger. And crucially, they don't just sit there quietly.
No, they are active. The sources describe them as spewing out toxic inflammatory sludge into their environment.
That is the SAT, the senescence-associated secretory phenotype. It's a mouthful, but it's probably the most critical concept in modern aging research. These zombie cells are constantly secreting a nasty cocktail of inflammatory cytokines, chemokines, and matrix-degrading enzymes. They're essentially broadcasting a "help me, I'm damaged" signal that never, ever turns off.
And this constant signal is incredibly damaging to the surrounding tissue.
It's devastating. It creates a chronic, low-grade inflammation that degrades the tissue around them. It breaks down collagen and elastin. But the worst part of all is that it acts like a contagion.
The rotten apple effect. One bad apple spoils the bunch.
That's the perfect analogy. If you have one senescent cell spewing this SAP sludge, it actually damages the healthy cells next to it. The inflammation it creates can trigger DNA damage in those healthy neighbors, and in some cases, it can actually push them into senescence too. It spreads the dysfunction through the tissue.
So if these zombies are a key driver of aging and inflammation, the obvious solution seems to be kill the zombies.
Enter senolytics. This is, without a doubt, one of the most exciting frontiers in medicine right now. Senolytics are a new class of drugs designed to selectively target and kill these senescent cells while leaving healthy, functioning cells completely alone.
And the sources we reviewed highlight a specific combination that's getting a lot of attention. D plus Q.
Dasatinib and kerosene. It is a really fascinating pairing. Dasatinib is actually a leukemia drug, a potent chemotherapy agent called a tyrosine kinase inhibitor, and quercetin is a natural flavonoid, a compound found in things like apples, onions, and capers.
So why do they work together, and why on earth do we need a leukemia drug to fight aging?
Because senescent cells are surprisingly tough. To avoid dying, they have to upregulate what are called survival pathways. The sources call them SCAPs, or senescent cell anti-apoptotic pathways. They've essentially turned on an invincibility shield to protect themselves from their own internal damage signals. Dasatinib works by targeting specific enzymes, like the Sangrar-C family kinases, that some types of senescent cells rely on to keep that shield up. Quercetin targets other survival pathways, like one involving PI3K. Together, they disable multiple parts of the survival mechanism at once.
So they're not killing the cell directly, they're just lowering its shield so the zombie cell's own self-destruct programming can finally kick in.
That is exactly what's happening. They allow the cell to finally undergo that apoptosis it should have undergone months or years earlier.
But the strategy for taking these drugs is completely different from, say, a daily blood pressure pill. It's what the researchers call a hit-and-run approach.
That is the key. You absolutely do not want to be on a leukemia drug every single day of your life. The sources detail a specific clinical trial that illustrates this perfectly. It's NCT 072700120, and it's focused on a condition called secondary progressive multiple sclerosis.
Let's dig into that trial. Why MS? What's the connection?
Well, in secondary progressive MS, there's a significant component of ongoing neurodegeneration and chronic inflammation that isn't really helped much by standard immune suppressing drugs. The hypothesis is that senescent cells in the nervous system, maybe the glial support cells, are accumulating and driving this chronic, smoldering decline via the SSP.
And the protocol they're using in that trial.
Intermittent dosing. In this specific trial, the participants take the D plus Q combination for just a couple of days to clear out the existing burden of senescent cells, and then they stop for 2 weeks or even a month before the next dose. The idea is to go in, act like a cleanup crew to wipe out the accumulated zombie cells, and then get out. You let the body heal and hopefully regenerate in the absence of that inflammatory SASP without the constant presence of the powerful drug.
That makes perfect sense. It's like weeding a garden. You go in and pull the weeds and you step back and let the flowers grow. You don't spray herbicide every single day.
Yeah.
But there's another approach mentioned in the stack, right? If killing the zombie cell is too risky, maybe in a fragile tissue like the heart or the brain, maybe we can just gag it.
That's the strategy behind a different class of drugs called xenomorphics. The root morph means to change. We aren't killing the cell, we're trying to change its behavior. We're trying to modulate that SASP2, as you said, tape its mouth shut so it stops screaming those inflammatory signals.
What kind of drugs can do that?
Rapamycin is the most famous and well-studied one. It inhibits a central metabolic pathway in the cell called mTOR, and by inhibiting mTOR, you can significantly dampen that secretory phenotype. Another class of drugs being explored is JK inhibitors, which are already used for autoimmune diseases. They directly block the signaling pathways that lead to the production of inflammatory cytokines.
So the cell stays alive, it's still there, but it stops being a bad neighbor.
Exactly, and this might be a much safer or preferable strategy in tissues where you just can't afford to lose any cells, even the damaged ones. Or in very elderly, frail patients where a mass die-off of a large number of senescent cells might be structurally destabilizing to a tissue. It's a more diplomatic approach to the zombie problem.
It's just incredible to see how fast this field is moving. We went from a vague idea of wear and tear to developing, molecular sniper rifles for zombie cells in just a few decades. But what does all this mean for us?
Yeah.
For society?
This brings us squarely into part 4. The implications. And I think the single biggest one is the shift in focus from lifespan to health span.
I hear this term a lot now. It's not just about living to be 100. It's about not spending the last 20 years of that life in a nursing home suffering from multiple diseases.
Precisely. Modern medicine is already very good at keeping us alive. We treat the heart attack, we manage the diabetes, we treat the cancer. But we tend to treat these as separate, isolated events. The field of geroscience, the study of aging itself, argues that we should be treating the root cause of all of them, the biological aging process.
The idea being that if we can slow down or reverse the accumulation of senescent cells, or better manage our oxidative stress, we delay all the diseases of aging at once.
The potential economic impact of that is just staggering. I mean, imagine delaying the average onset of Alzheimer's, cardiovascular disease, and frailty by just 5 years. The savings in healthcare costs, and more importantly, the gain in quality of life, would be astronomical. It would completely change the structure of our society.
But it's going to get very personal, isn't it? We talked earlier about how the one-size-fits-all antioxidant supplements failed because patient heterogeneity.
Yes. The future of this rust management will have to be highly personalized. I can easily see a future where, as part of your annual physical, you get your oxidative balance score measured.
Like a credit score, but for the health of your cells.
Exactly. And the prescription won't be a generic go take some vitamin C. For some people who maybe have a genetic weakness in their endogenous defenses, maybe they do need specific antioxidants. But for others, like someone undergoing cancer therapy, where we actually want high levels of oxidative stress to kill the tumor, we might even prescribe pro-oxidants. Or for the elite athlete, we might precisely time their nutrition to make absolutely sure we aren't blunting that beneficial hormetic signal from their training.
It's precision medicine, but applied to the problem of rust.
And we absolutely cannot ignore the environmental implication here. We started this whole conversation talking about plastics. The oxidative degradation of our material world is the mirror image of our biological degradation.
The microplastics entering the food chain.
Exactly. That oxidized plastic debris, that chalk, it carries free radicals. It carries adsorbed toxins. It enters the food chain, and ultimately it enters our bodies, potentially adding to our own internal oxidative load. So solving this rust problem isn't just about finding better drugs for humans, it's also about better material science. We need to develop better UV stabilizers that keep plastics from chalking and shattering into that microdust. Or, conversely, we need to design new materials that degrade cleanly and biologically using enzymes rather than oxidatively shattering into a persistent pollutant.
It connects everything. The health of our individual cells and the health of our planet are, in a way, fighting the very same chemical battle against entropy and oxidation.
It is a profound reminder that we are part of the physical world. The laws of thermodynamics apply to us just as much as they apply to a plastic lawn chair. But the difference is we have the intelligence to understand the process and perhaps to intelligently intervene.
So we're walking this incredible tightrope. On one side, we have rust, the unavoidable damage that accumulates from the act of living. And on the other side, we have signaling the necessary beneficial stress that keeps us strong and resilient.
And the goal isn't to eliminate stress or radicals entirely. That would be a disaster. The goal is to maintain the balance, to keep the signal clear and the noise down, to manage the zombies without destroying the town. It's a nuanced, incredibly complex dance, but for the first time, we are really beginning to learn the steps.
And that, I think, is where it gets really interesting, because as we learn to precisely manipulate these fundamental levers, senolytics, xenomorphics, personalized redox management, we might be on the verge of redefining what it even means to get old.
Indeed. We are moving from simply observing the decline to actively managing the biology of time itself.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content. And support us on Patreon.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.